Welcome back, everybody. This is the Principles of the Innate and Adaptive Immunity, Chapter 1.5 on Antigen Binding. Please make sure to subscribe and follow along because this is the fifth lecture of a longer series of all things immunobiology. And please make sure to like and also comment below on what things you'd like to see, what things could be improved, and also what other things that you'd like me to clarify on if you have any questions. So let's begin. This is epitope immunobiology. So an antigen is a toxin or other foreign substances, okay, that induce an immune response in the body. Okay, so what I have here is a typical, for example, it could be bacteria cells with some flagellum on the outside. Okay, and essentially an antigen could be anything that could be secreted out of the bacteria or pathogen. It could be anything that's molecular signatures on the surface and also on the flagellum that's sticking out. So I see here I have a few other diagrams here. You got like a triangle and a circle. These essentially to our bodies or our innate immune system would be pathogen associated molecular patterns as I covered in the previous lecture. And these would essentially be non-self, okay? Non-self meaning they don't exist in the human body, but they do exist on our pathogens. So this is what can be detected by our dendritic cells, our macrophages, or essentially our innate immune responses to say that there's some foreign pathogen that's living inside of our cells now, invasion. Okay, and what these are called is an epitope. Okay, essentially is a molecular structure of an antigen molecule, which is known as an antigenic determinant or an epitope. Okay, essentially, it's certain areas of these moieties that can be detected by our proteins. Okay, so by proteins meaning our T cell receptors and our B cell receptors. This is essentially where it will be binding to. Okay, the epitope is the binding spot of our T cell and B cell receptors in our innate immune responses. Such things include certain proteins that we don't that we don't code for example, or glycoproteins, or polysaccharides, of pathogens. These are common ones. These macromolecular antigens usually have many different epitopes, okay, that can be recognized by different antigen receptors. Okay, so for example, if I'm just going to draw a hypothetical epitope, and let's just say there's on the top here is a triangle, and in the middle part is a circle, and somewhere near the bottom of it has a square or a rectangle of some sort. The epitope could be this portion right here, or it could be this portion right here, or this portion right here. Maybe it's a combination of the two as well. Okay, so essentially, this is the antigen, and the epitope could be anywhere on here. Essentially, T cell receptors, B cell receptors could detect any pieces on here. Okay, so let's say, for example, we have these antibodies, or we can hypothetically say these are B cell receptors as well. Okay, that are anchored to an B cell, and they're going to be surveying the areas of infection, or just essentially all the tissues for foreign pathogen invasions. So you see, got an antibody here or B cell receptor, and they go in and they happen to bounce off of these two moieties or possible epitopes. It doesn't bind anything, so it isn't detected as a foreign. But we also have other B cell receptors or T cell receptors as well. I'm just going to use a B cell receptor, for example, here, that could possibly be binding. So this one might bind the circle piece, and this one might bind the triangle, okay? And because essentially once these are bound, then we know that this piece here is foreign, and therefore we can then ingest it. For example, the macrophage or the dendritic cell can then ingest this bacterium or this part of the flagellum at least, and then of course present it to the adaptive immunity. And so therefore we need to talk about a few things. The antigen recognition. Okay, so for the B cells, recognition is directly recognized. It directly recognizes the native antigen. Okay, so this is very, very, very key. The B cells detect the native antigen. So, for example, that is a pathogen or the protein itself that comes off. That is of the antigen. Okay. So again, this will be secreted or expressed on the surface. As a result, the B cell will differentiate into effector plasma cells to secrete antibodies that will bind to and neutralize these antigens and pathogens. Okay, so we talked about that in previous lectures. But again, to summarize this top part, the B cell binds to the native antigen. Okay, so that's again like full bacterium or maybe just a protein that's been secreted off. And the result, the B cells then become the effector B cells, which essentially are plasma cells to secrete antibodies that can then all bind to that piece of native antigen that we noticed earlier. That they recognized earlier. For the T cell, however, there's a slight difference. It recognizes processed antigens. Okay, so not native antigens. It recognizes processed antigens, and they're partly degraded peptides displayed on the surface of antigen-presenting cells via MHC complex presentation. So this is a major histocompatibility complex. It's right below here. Okay, we'll talk about that later. But essentially, the T cells only recognize processed, okay, and degraded peptides. Okay, and essentially what it results into is that the T cells then differentiate in effector T cells depending on the type of MHC complex. So there's a few different types of MHC complexes, and we'll talk about that in another lecture. But depending on which type, then it can help it differentiate into helper T cells, cytotoxic T cells, and regulatory T cells as well. Again, those are the three types we mentioned in the previous lectures. But what this means is that essentially these macrophages and dendritic cells, after engulfing these proteins or these pathogens, it degrades it all up, and the little pieces of it then gets expressed out on the end of what we call an MHC complex, the major histocompatibility complex. And then these are required to be processed and then presented to the T cells. Major histocompatibility complex, a set of cell surface proteins essential for adaptive immunity or immune system to recognize foreign molecules. Okay, so I'll show you what that looks like here. So MHC class presentation, we have, let's say this is an entire pathogen or antigen that was expressed and secreted, and essentially the pathogen, the dendritic cell, recognized this piece and engulfed it. Okay, and let's say this red piece is essentially the piece where the T cell will eventually recognize, because the T cell receptor happened to be able to bind this piece. After being engulfed by the macrophage or the dendritic cell, then it goes to the cytotoxic, it gets degraded by degradative enzymes, toxic degradative enzymes in the cell into little pieces, and then it'll grab all these pieces and actually express it on the cell surface through an MHC complex. And for example, it grabs this one red piece, and then it'll actually express this on the MHC complex. Okay, so you see this is now called epitope peptide MHC. The epitope binds to the MHC complex, and then of course this is the antigen presenting cell right here on the below, and then it actually presents it to the T cell, and the T cell will survey all the ones that are on the surface, and I see that okay, it happens to bind this epitope, and now this T cell receptor can be signaled for survival and clonal expansion and so on. Okay. Expansion. All right. So essentially what the T cell receptors require is an MHC complex interaction, whereas the B cell receptors don't. They can bind directly to the pathogens. And for the binding and survival, this is also very, very key, because it is inevitable that potentially dangerous receptors can react against an individual's own self-antigens, okay, that will be produced. So essentially, these T cell receptors, when they're made, have a lot of variety at the ends here. This piece right here, and this piece right here. Essentially, they'll bind to all different types of things, and that includes ourselves, and that's called autoreactive T cell receptors. We don't want that. So what happens is that when they're actually being developed, the T cell binding, for example, it binds to an epitope. It binds very, very strong. That's why I have a very large font here. They'll die. If it binds very, very, very lightly, it also dies. But it happens to bind just perfectly, just right. You can just say it's kind of like Little Red Riding Hood, where there's not too much or not too little of binding. This would signal clonal expansion. So I'm just going to put CE here, okay? CE, clonal expansion. If it happens to bind self-binding very, very strongly, mediocrely, and also very, very minimally, all of these will signal for cell death. Okay, so essentially programmed cell death is apoptosis. So if this T cell receptor happen to bind self-antigens or self-epitopes, they will be signaled to basically go through apoptosis and die. And so you see how the only possibility here in green, which is process for clonal expansion, is that with binding mediocrely well, pretty well actually, to foreign antigens. If you bind too much or little, they'll die, and also if you bind any self-antigens, they'll die. Okay, and this applies the same for B cell receptors. So in summary, the B cell receptors and antibodies bind to native antigens and pathogens. So this is very, very key, native pieces, okay? T cell receptors bind to partly degraded antigens that are presented on the MHC complex of antigen-presenting cells. So again, this is partly degraded complexes, antigens. Binding of the B cell receptors and T cell receptors to foreign pathogens has to be just right. Okay, think about the Little Red Riding Hood again. It has to be just right in order to signal clonal expansion. Binding to self-antigens of any sort by the B cell receptors or T cell receptors results in apoptosis, essentially cell death. Okay, so this concludes up our chapter 1.5 on the B cell and T cell receptors. Please make sure to subscribe for following lectures in all things immunobiology, as next time we'll talk about the lymph node anatomy and how T cells mature in here, as I briefly scraped over that in the previous lectures. Now I want to go through this in more greater detail and how the innate and adaptive immunity really connect together. So again, make sure to subscribe and follow along, and like this video if you haven't if you happen to be getting something out of these videos. And of course, let me know in the comments if you like the videos, don't like the videos as well, or is there anything in particular that you want me to clarify, and I'll follow up. So thank you for following on. This is Epitope Immunobiology, and I'll see you next time.